CRISPR-Cas9 is a method of targeted genome editing using clustered, regularly interspaced palindromic repeats and an RNA-guided nuclease. Initially isolated from bacteria, CRISPR-Cas9 is an upgrade on previous methods, as the old utilized protein-DNA interactions, whilst CRISPR-Cas9 is able to utilize simple base pairing rules. CRISPR-Cas9 is able to introduce double-stranded breaks at a particular sequence and requires the guide RNA and Cas9 protein to be expressed in the organism. The guide RNA consists of a 20 nucleotide sequence at the 5' end of the strand, which directs the Cas9 complex to the target site. This site must be directly adjacent to a PAM, or a protospacer adjacent motif. Nuclease-induced double-stranded breaks can be repaired by two methods: non-homologous end joining or homologous directed repair. If a template is included, homologous repair is more likely to occur, resulting in no loss of genetic information.
Cancer at its simplest form is when a cell loses its ability to follow the body's control mechanisms responsible for maintaining a normal growth rate. The human body is composed of billions of individual cells, all following these strict rules for growing and dividing. However, when the genetic material called DNA inside the nucleus of each cell responsible for cell division becomes damaged and consequently unable to repair itself, the cell grows and divides uncontrollably. When a cell reaches this point, it's classified as a cancer cell. Its classification is then further refined into being benign or a malignant tumour. Benign tumours are localised and do not spread to the rest of the body. However, if left untreated, they can grow in size and become detrimental to the health of the individual. Whereas malignant tumours are a form of cancer that spreads throughout the body via the bloodstream into adjacent organs and tissues, which can be life-threatening.
CRISPR-Cas9 is able to induce double-stranded breaks at defined positions using two catalytic centers, also known as blades. These blades cleave each strand of DNA target sequence next to a PAM sequence and matching the 20 nucleotide sequence of the single guide RNA. This single guide RNA contains a dual RNA sequence taken from CRISPR RNA and a separate transcript that binds and stabilizes the Cas9 protein. This Cas9 single guide RNA-mediated DNA produces a blunt double-stranded break that triggers repair enzymes to disrupt DNA sequences at or near the double-stranded break site. Inactive forms of Cas9 can then be used to regulate transcription. This ability of the Cas9 enzyme to induce double-stranded breaks has become an interest for scientists as a possible cancer treatment. CRISPR-Cas9 has already been established as an improved method to generate liver cancer in mice. This robust technology for rearranging genomic sequences provides development and progression in the field of cancer research and treatment.
While CRISPR-Cas9 has shown that it is an effective tool in genome editing for research, it does, however, have some limitations for its application to cancer treatment. One of the challenges involves efficiently delivering the CRISPR-Cas9 machinery into the cell. Plasmids, viruses, mRNA, and Cas9 ribonuclear proteins have been used in the delivery. Ribonuclear proteins are able to deliver the machinery into the cell that were previously difficult to transfect. However, they can only be used in vitro as their delivery efficiency in vivo is significantly reduced. If this delivery method was used in cancer treatment, not all cancerous cells would process the CRISPR-Cas9 machinery required for the genome repair. Lentiviruses can target a variety of cells, but they are capable of causing further mutations, especially if they are used alongside a designed nuclease. Adenoviruses are most promising for viral delivery as they can edit genome with greater specificity. However, they can only carry 4.7 kilobases of DNA and are not as effective when used in vivo. There is also the risk of CRISPR-Cas9 machinery causing off-target changes due to the site being homologous to the target site. These unwanted changes could have a variety of consequences. If the CRISPR-Cas9 system were to be used as a future cancer treatment, an effective delivery system would need to be established. As well as this, the target site would need to be carefully selected to prevent off-target editing.
Caution flags have been raised about the use of CRISPR-Cas9 on human germline editing. This research has generated the debate among world-renowned scientists about the ethical concerns and implications of CRISPR-Cas9. While some members of the scientific community have argued that a delay should be put on human germline editing, others have argued that it is unethical to withhold a technology that would eliminate devastating diseases. These arguments show that ethics regarding CRISPR technology need to be addressed when being implemented into society.
These arguments show that ethics regarding CRISPR technology needs to be addressed when being implemented into society. One of the risks of this treatment is the introduction of alleles that could potentially have unforeseen side effects on the genome and phenotype of an individual. A regulatory institute needs to oversee human genome modification projects to ensure safety and ethical concerns are addressed. An example that could be seen as potentially unethical is the proposal of designer babies. This is a term given to babies that could potentially be born with modified and predetermined genes. This is an ethical concern for society. Many believe that heading down this path in human genetics is against nature and has many religious implications.